Well, agriculture faculty, I think you have. Could you hear me now?
Good morning, sir. Yes, sir.
Okay, good morning. Good morning. So all of you are welcome to the Faculty of Agriculture already. I missed your orientation also. I was somewhere else. And today now, I think we are seeing here, this is myself. I think we later will meet. And I am, I think you know, Dr. Kumaran, Professor in Agriculture Biology. And he is myself, he is the head of the department, Dr. Bahiraran, and others are there. You know, I think you are also familiar with that one. Just go, sorry, just a moment. Okay, here, this is myself. I think, okay, this is the Vice Chancellor. And the former Dean, this is the present Dean. And you are head of the department, yeah. Okay, yes, you are head of the department of biology. I'm the professor in agriculture biology, Dr. Kumar. It's okay, okay. Welcome to for this session. Okay, experts are there. Okay, anyway, this is our post. Don't worry about today's success. Just interaction. You are welcome to agriculture, and at the meantime, to move on that one. So in this course, with agriculture biology, you know very well, there are 6 departments, and from your department, our because cell biology and crop property, we are going to study cell biology and crop property. This is AB, 1 note 1 2. These notations and everything you know very well. Okay. Here in this, worry about that one, I have sent to red result. Amina, we have already seen that 2 documents. First one is virus, the other one is
Bacteria.
Bacteria. Good. So please cooperate. Okay, that's good. You can anytime again, if you can hold me, ask me, chat me, you can put any doubt, you can put it in the chat box. Isn't it? Samina Jayakumar. Okay, we will move. Here, don't worry about that's what bacteria, just look at read it and virus. No need to that much. That's I have said it, but here we study the virus. That's the 2 purposes. One is to understand the basic of the organism, and there is some beneficial bacteria also there. Especially we are getting for the genetic engineering, but it's okay. Anyway, the first one we study is here. We are going to later. This is the basic. We are going to study the biotechnology and the professor of the biotechnology. Biotechnology is include the agriculture biotechnology, plant biotechnology, including the genetic engineering. Genetic engineering also is important. Yeah.
Your voice is not clear.
Oh, really? Okay, thank you. That's great. Others, but just I will choose 10 just click. Students, okay. Just what about now? Is it, can you hear clearly, or is still this problem? Anyone, Samina? Could you hear me clearly?
No, sir.
It's not clear. That's the internet problem. Everything is like usually I take the lectures early morning 6 o'clock and 8 because of this internet. But your voices are clear. Why is still I'm using the headphone? Samina, once again, please tell me. Is it now okay or not?
No, sir.
Okay, just wait. Everyone, please wait. Now, the problem on the Hello, dear students. Could you hear me now? Is it clear?
Yes, sir.
Okay, that's the internet problem. When it's go for the Wi-Fi, because at the home it's three people. Yeah, at the time we are using the room. So I use the Wi-Fi. It's a problem. It's okay. Thank you. Okay, thank you. Okay, now I will come. Even still the video also not clear. I will put it. I think it's not clear. Just one moment. It's okay. When you start there, maybe there's some problem. I will be. It's not coming. Okay. So could you see my videos?
Yes, sir. Yes, sir.
Okay. Then we'll go. Okay, sorry. Okay, here we are going to study the cell biology and crop botany. Just wait. Biology, because our ultimate aim is I am the professor of the biotechnology. We'll study the theory of a stem cell, we have the biotechnology, and we do the genetics and plant breeding. And the for one, when you choose the specialization, plant biotechnology and genetic engineering. So, commonly we can say biotechnology. I will teach you the biotechnology. So, what is biotechnology? Biotechnology is simple. So I think you have heard about a lot of things about the biotechnology. But it's simple. If you everything is techniques. Even what you do is if you boil the milk and how to cool down is a technique. Before after buying bring your milk at home. Before you drink the milk, you have to boil it. So boiling is one of technique. You have to eat something. Even the curd production. It's an ancient, modern, old bad technology is curd production. So in the curd production, you know curd production. Oh, even your grandma is specialist in the curd production. They will bring the milk and boil the milk, allow to cool down. Each and everything is a techniques. Boil the milk, allow to cool down, then mix with the previous culture. You will use mostly they don't have previous one. They will go for the neighbor and bring some spoon of previous culture, put it there, and overnight they will allow. Next day it will produce the curd. So what is biotechnology? If you use everything is technology, but if you use some if you use any organism in that technology, that is biotechnology. Bio means organism. If you used organism, that is biotechnology. Even the herd product, they are using the herd. In the even the bread bakery, bread production, even the yogurt. I think you have heard about yogurt production, wine production, cheese production. Each and everywhere they are using the organism. So that's a biotechnology. So technology with organism. Bio means biotechnology. That's what we are going to study later. If you go, so I will teach you what is bio. First, we want to understand what is organism. Biotechnology means bio means organism. We use organism in the techniques. All up that comes under the biotechnology. So what is organism? Living organism. Bio means living organism. Any living organism we use, that is bio and technology, biotechnology. So we are going to study living organism. So now don't worry about the slides. That's today's listen. So living organism. So we are going to start from the ancient living organism, very simple living organism. But what is non-living organism? We don't care about that part because we are also going to be using the living organism. So we will start from this living organism, where from the very oldest and smallest cell. Isn't it? That's good. Okay, welcome. So we are going to study the living organism. Living organism, the oldest organism, one of this kingdom of the bacteria. So we will start from the bacteria. That is to understand the basic units of cell. Then thereafter we will study from the bacteria, kingdom of, then we will come from the kingdom animal, then kingdom plant. So we will always use, as agriculture students, we are going to use the plants. So the basic, we have to understand the basic of the plant, that is cell. Cell. What is the ancient cell or oldest and simplest cell is bacteria. So first we will study the bacteria, we will understand the basic units of cell, then we will come to the plant cell. We will study the plant cell. Why we are studying the plant cell? So plant cells, the basic units of the plants. Plant cells together, they will form cells together, they will form tissue. Then tissues, they together form organ. An organ, if an organ together, they will form system. Then system together, they will produce organism, living organism, human being. Understand? Cell together tissue, tissues together organ, organs together system together whole organism. So we will go start from the plant cell. Especially within the cell, there's a nucleus are there in the center. Within that, there's a DNA are there, DNA, RNA. Then later we are going to study about the genetic engineering, how to transfer the gene from here to there. Isn't it? That is genetic engineering. So before that, we should understand the basic unit of plant cell, that is cell biology. In the cell, if you want to understand the basic of the oldest cell, that is kingdom monada, that is bacteria we are going to study today. That is, we will start the living organism. First living organism in this world, we think bacteria. Others are non-living. What about virus? I think now everybody has virus. Is the most popular name now in the world, this virus. Especially they will name it Corona and COVID-19 pandemic. It's become a next speech before 2019. Nobody don't worry about that virus. But anyway, I don't like to start from actually we have to start from this virus, but I will start from the bacteria. Next class we will see about the virus. Why we are studying the virus? We are going to study the living organism. So if you want to understand the basic units of living organism, we will go for the bacteria. It's easy to understand. If you want to go for the mathematics, first you will start with the numerical numbers 1, 2, 3, 4, 5, 6. Isn't it? Like that one. If you want to go for the genetic engineering or biotechnology, you have to study the cell biology. So if you want to go for the plant cell, before that, understand the bacteria cell. So this is bacteria start from the living organism. Others are non-living organism. So virus is where it's located, whether it's living organism or non-living organism. If you ask really, you can't categorize the virus under living organism because there's a except multiplication or reproduction. They don't have any other features that could be compared to the living organism. But you can't categorize the virus in the non-living organism because it can multiply. It can reproduce. Same multiplication, division. Or only the virus and bacteria, they will divide it. One bacteria will divide into two. Next class we will study. To divide into two and multiply. They will multiply. Only this in the mathematics, division and multiplication is totally different. But in the biology, division and multiplication, the production is doing the same meaning. When it divides and it becomes multiply, the virus is doing the same. They multiply very fast. So they can easily viral. So virus, you can't categorize under living organism because it can reproduce. Only single character. You can't categorize that this virus as a living organism because there are no other characters. You can't compare with the living organism. So only it has the character with that's the living organism has, that is, it can reproduce. So we will learn virus also. Next class we will study. Then actually from the bacteria we can start to understand this one. So here you can see we are going to study the kingdom monoda. I want you to understand, otherwise fungi, animalia and plant kingdom will come later. So kingdom monoda. So cells have evolved two different architecture. There's a prokaryotic style, then eukaryotic style. It is better, my dear students, please write. Don't think why you are going to write. There are some tubes are there. I can be easy. You work hard, definitely you will get fruits. But if you do lazy and you to say some reason and that's a problem, I have the phone. Telling the reason is very simple. You work hard, but don't waste your time. Don't blame any other situation or blame anybody. It's your duty at the moment, but it's your duty. Slowly we have to introduce. practice your English. But usually, including myself, when I come to first year, first semester, I also had a very poor in English because that time we didn't worry about the English. That time everybody was saying, "Oh, you are going to be here, then no need." Later on, we understand after enter the university, the first year, first semester, very poor man. But later I didn't try anything to learn English, but it's automatically been improved. And even I did my PhD in the Tokyo, Japan, and I even improved PhD and I became a professor and everything. So no problem. Don't worry about that. But, so this bacteria, these are the prokaryotic and the unicellular. Bacteria have cell wall and it has circular DNA called plasmids. And there may be aerobes or anaerobes. Some of the bacteria, they can survive in the when the air is present. Some of the area, they survive where the air is absent. That is anaerobes and aerobes. Some of the bacteria, they are heterotrophs or autotrophs. Heterotroph means we study. They will produce the energy. And anyway, bacteria are awesome. So why we are studying is it that much of important? Earlier they have we are senior when I teach the bacteria virus first class, they have viruses. Why do you want to study virus? It's what's the importance of virus is studying as a first class unless you are starting from the virus. Why? But now everybody are talking about the virus. Even if you ask your grandma or grandfather, they can tell who listen the radio or watching the TV news or reading the newspapers, they can say a lot of stories about the corona. How it started, how it spread, what's the, how can you prevent and everything. Yeah. Your younger child, the sister, they can tell about this bacteria. Okay, because bacteria are very awesome, very important. Even the bacteria, there's bacteria we can't survive in the world. The first oxygen produced in this earth by the bacteria, cyanobacteria. So that much of bacteria are important. So if you try the small points, even I have told I don't teach in example interview, but I can say something. So even the exam, you can write bacteria, they are the prokaryotic and unicellular. They have cell wall, they have circular DNA called plasmids. We will study this important circular DNA called plasmids. The genetic engineering we will study. They can be aerobes or some of they are anaerobes. They can be heterotroph or autotroph. So these are the simple characteristics we are going to discuss about this one. So where is bacteria? It can be present in each and everywhere. They can found almost everywhere. Where can be found? They can be in the water, the waterborne pathogens. They are some air, airborne pathogens, and the soil-borne pathogens, food pathogens, even the skin or inside the body, each and everywhere on the most object. You can see the bacteria. They are the so, but what's the difference between the virus? Bacteria can be anywhere, but viruses, they can only survive within a living organism, another living organism. Even your saliva, it has organism, it's a living organism. So, virus is started through that saliva. That's what we are having the face mask. When you speak, you have infected and you are the vector. When you speak, it will spread through your saliva. So even if you touch, you don't touch the saliva because it will easily enter. That's what we are using the mask and everything. So this one bacteria, don't worry about that. I have sent the tube. You can go through that one. But anyway, through this tube, it's okay. You write something. You have to practice your pain. Otherwise, you can't write it for your exam. You don't write you the English and everything. Why we are teaching this one again? Because we have to write and practice the spelling and simple sentence and everything. So please follow that. So this is bacteria everywhere. You can see and don't worry about much of it, but this is bacteria is very important. The world. There's no bacteria in this world. What will happen? There's a lot of beneficial bacteria. Even they are the decomposers. They are break down our garbage. Otherwise, there's no bacteria. There's no way to degrade our garbage. What will happen? Entire world will fulfill with the garbage. But the bacteria, they will degrade that garbage and break down this organic and bring to the inorganic and available to the plants to take. So bacteria, they are doing a lot of things. Even bread making, cheese making, yogurt making, everywhere, bacteria. Use the bacteria. So there's a lot of beneficial bacteria also there. So you can see the simple prokaryotic bacteria, simple as an oldest form of the life, usually single cells. You can go for the letter also. You can just note it that one. Okay. This one is okay. Why we are studying the cell biology? We will start from the bacteria and we will go later also. Okay. Now my question is, okay, anyway, okay. This is about the cell biology. You can think about that one. So why we are studying the crop botany? In this lesson, most probably during the lectures hours, I will take cell biology, and mostly the practical side will take up about the crop botany. Why we are studying the crop botany? I think last class we have been there, the practice. In the crop botany, what we are going to study? We are going to study the families. Within the families, the genus. Within the genus, the species. So what are the families we are going to study? I think we have heard about Fabaceae, Poaceae, Solanaceae, Cucurbitaceae, all these families we are going to study. Within the family, even for example, Solanaceae. Yes? Did you study? Last class, have you started? Last class, have you started? Samina?
Yes, sir.
Okay.
We studied.
Okay, that's classification. You will understand that. What is the classification? Thereafter, you will come down up to the family. Thereafter, family. So, for example, you will have Fabaceae. All the pulses. You can see the pulses. The green gram, black gram, all these pulses comes under the Fabaceae. You take the family Poaceae. All the grains. Rice, barley, wheat, and all these things of the Poaceae. You take the Solanaceae, that is potato, tomato, brinjal, or tobacco, all this that comes under the family Solanaceae. Within that, you will see the genus. Okay. Solanum tuberosa is a potato. Isn't it? Each and every you learn there. So why we are the crop botany, what we are going to study? We will study each and every part of the crop. Simply, if you want to say, how to describe the angiosperm. How to describe. If you take the one leaf, how you take one of the leaf from your home and describe now, you will say it's a green color. That's all. But after studying this one, you can describe more than one page. The shape, the size, the tip, venation inside each and every other edges and everything. You may have one page. You can now you bring one leaf and ask me what is it. Describe this leaf. You simple. You say it's a green color leaf. But after studying this, we can able to. So why we are studying? the purpose, because later you will produce a new crop. You want to say exactly new variety. Then thereafter you are able to describe the new one. We study in the third year, second semester, genetics and plant breeding. Genetics and plant breeding. Okay. You can, if you want, you can see the tube over later also. Okay. Just listen. We will study the third concepts of plant breeding, genetics and plant breeding. In the plant breeding, we will study how to develop the new variety. How they develop new varieties. What is variety? What is cultivar? You know? Okay, I will come. So what is variety? Anyone tell me one example of variety? Hello? Are you there? Anyone? Can you say what is variety? Samina?
Different types.
Example. In the plant variety. Variety means you have when you go for the choose a dress in the shop, saris or something, so salwar or what skirt or blouse, you have to do lot of varieties. You will search so many varieties. After searching the 1,000 variety, you will select one. It's okay. The plant variety. Okay, thank you, Samina. In plant variety, variety, simple. Cultivar variety, the plant variety. Okay, thank you. Anyway, it's okay. You will learn each and everything, but after reaching the final year, you will say, we didn't learn anything from the agriculture faculty. Okay, it's okay. Variety means simply, okay, if I ask you, you will tell simply. In the mango, what different mango you have tasted so far? Mango. Just talk. Kartakolumba, Salem, Ambalavi is different. Each and every, these are varieties. Villat is one variety. Within the mango, one variety. The Kartakolumba is another variety. Isn't it? Salem is another variety. What about the banana? You have different varieties. Isn't it? Kappal or Kolikuru or Kadali or either or something else different. These are varieties. For all the crops, it has lot of variety. To take rice. Okay, rice, can you say anyone rice variety?
Kora.
Sorry?
Kora.
Rice variety.
Samba.
If you ask
Samba. Red rice.
Okay, not red. No. Red rice is not variety. If you ask your grandma, she will tell more than 100 varieties, even the old variety. Just simple. Atagari, Basmati, Samba, Kiri Chamba, these other varieties, each and every one. Earlier, the Motagarupan and all these other varieties. All these varieties, okay. Please mute others, please. Okay. So just please mute. Okay, thank you. So all these varieties, these other varieties, all these varieties have developed. Okay, thank you. You mute. Please, you will study. Hello, mute. All these varieties has been developed. All these varieties have developed by conventional plant breeding. Plant breeding. Who developed this variety? Plant breeders. Plant breeders. You can become a plant breeder after this study. When you come to you will learn in the 32nd semester genetics and plant breeding. If you choose the agriculture biology as your specialization in the final year, you can do your final year research in plant breeding, and in future you can become a plant breeder who can develop new variety. If you want variety, if you think the Vilat is very good color, very good the red color and yellow color. But the taste is comfortable one is better. The flavor and the taste of comfortable one is good. Am I right? But the comfortable one, the color is not attractive. It's not mostly green and little. If you prefer to do it should be the mango should be like a Karthakolamban shape or other oval shape, easy to peel, and very good attractive color, like look like a black color, the red, orange, yellow. But inside the taste should be as a Karthakolamban. So you have decided you have drawn the objective. That's the objective is, and the skin also very created flower of the Karthakolamban. There is artificial processing and pollination. Then it will become, within that one we can select which have like a Karthakolamban taste and the color of the plant. So you can develop, you can become a plant breeder, and after that you can develop a new variety like a Karthakolamban and that will mix and produce new one. Latakolamban, you can name it. So in that case, after producing it, you can able to describe the new crop. Okay, this is you can understand, no? This is there's a base capsule, which is a cell wall, this is rhizosome or nuclei or flagella or pili or cytoplasm. So you can throw it, you can understand. It's not serious thing. So what is capsule? Where it's located? You can see this is a capsule. Okay, I will come. You can notice that. So in the plant breeding, you will develop a new variety. After that, but you have to describe from root tip to meristem. Each and everywhere you have to describe, isn't it? To describe that and you need the property knowledge. Okay, now can you differentiate plant and animal? Okay. Can you differentiate bacteria and plant? Yes. Isn't it? You can easily understand which is animal, which is plant. Okay. Within the animals, you know, dog, cat, snake, elephant, you can simply identify. Am I right? There's no doubt you can understand, identify. Okay. Then even within a human, so if you take human, you can identify simply which is human. It's a man or human. So within the human, there's a lot of people are there. Can you identify each and everything? Yes. Who is Samina? Who is Janani? Who is Darsana? You can, all these people, humans, are look like exactly same. All of your batchmates, the girls, they have the exactly one head, two hands, within the hands, five fingers, the legs, five fingers, five fingers. Each and everything is look like same. There is no need, no difference. sometimes color also everything. But you can easily identify who is Amina, who is Janani, who is Dasana, and who is each and everyone. You can easily identify. Am I right? How? Same. The same as like even the you can able to have that much knowledge, but that much knowledge you should gain even the plant wise, and you will feel very difficult. But within a human being, exactly same, but you even 1,000 people you can remember. If I show Barack Obama, you will suddenly say even our within a people. You can able to identify exactly look like same. The same knowledge you should have in the plants as agriculture student, and especially we want to become a plant breeder. This is capsule, okay? There. So within the plants, you can identify easily which is mango, which is banana. Am I right? Which is rice. Some of the maybe few of students they don't know, they haven't seen the rice. They haven't seen the wheat. You haven't seen the potato plant or tomato or most of the things because they have born in Colombo or somewhere else. They were there. They haven't a chance to see this one. But other people they know. You say the mango, you say yes, mango. This is neem. This is banana. My question is within the mango, can you identify which is Kartaparama, which is Bilato, which is Ambalavi? Sometimes few students maybe say yes, I can differentiate. Who have at home, their home or neighbor's home, Vilata and Kartaparama, you can be able to identify. You can say this is Kartaparama, this is Vilata. But now my question is, can you write in the paper about their differences? What's the difference between them? How to identify that? If you're the person who don't know the Kartaparama and Vilata, can you describe the things about the Kartaparama and he should be able to identify. Not only the fruit, without fruit also they can able to identify. Can you differentiate Kartaparama and Vilar? Can you differentiate the banana, kappal and kadal, so called guru and kadal? Can you differentiate? It look like exactly same, but it's totally different. Am I right? This is okay, capsule. Keeps the cell from the drying out and helps it stick to food or that will help the capsule. Even the corona, because of the like a capsule like style structure that help to easily attach with others and easily we will take it. So that this one here we call capsule. That is easy to touch and keeps a cell from the drying out and helps to stick to food. Then that's corona that easily stick and transfer. Okay. So this is next importance is the bacteria, the cell wall. You can see that cell wall. Why what's important? The cell wall is important. Later you will study. We will come the plant breeding. Why we are studying the property. So you see the Karta Kulban and Vlad. There's no fruit, but you show the fruit you can maybe identify. By the plant without fruits, can you describe from top to bottom of the Vlad? Okay, you can take up one paper, full scrap sheet paper, write Karta Kulban. Then you write, describe from top to bottom, from the root and the tip and each and describe. And another take paper, you have to put Willard and you have to describe. There should be, there's total difference there. But at that moment, the knowledge, if you write about the banana, Willard, you like both are same. But there's clear difference. So that's what we are going to study the crop botany, how to describe, how to differentiate. So we take the banana, even Kadalian, couple or molecule. But very few students, they can see this banana and the banana fruits, and they can say this is very good. And even the most of the half of the students, they don't know even. They can't say by fruits, banana fruit, this is Kartapadambano, they have that much of. But without that fruit, you will see the banana, maybe out of this 135, maybe 5 students, they can able to identify the banana in the field without bunch. Maybe the 5 students, maybe they know which one is Kadaliyo, which one Kartapadambano. Other 130, they can't do it. You can ask me, can you differentiate which one is Kadaliyo without a bunch? In the banana field, you go, which is Kadaliyo, which is Mondan, which is Polito. No, I am very sure. More than 130 students, they don't know. So that's what we are going to study, because later you are going to develop a new variety. You can become a plant breeder. That's what this my correct in my subject biotechnology. We will learn how to improve the genetic material of the crop. Crop improvement. Simply, I will teach crop improvement genetically. Genetic material, how to develop new variety through the conventional plant breeding, at the same time through the genetic engineering. The latest technique, the great technique, we will learn later. I am not going to go give each and everything to the next day. So that's what we will develop a new variety. Then thereafter, after developing the new variety, for example, I have told Kartapalman and Milan, we mix together, we can produce another Platapadumban. Or anything, you can name it. Then you have to describe your new crop that could be differentiated from the Virat and Kartapadumban. You could differentiate. After your description, you have to describe your plant, new plant, in the 10 pages. So who see that one, they can able to understand, yes, this is totally different from Virat. This plant is totally different from the Kartapadumban. You know, Kartapadumban and Virat is totally different, no? But if you look at the mango tree, it's very difficult to differentiate. But after studying this properly, you will understand. That's what we are studying the property. Okay. So what is the ultimate purpose of my course? Crop improvement, biotechnology. We will improve the crop genetic material. So what will we do? We will develop the new variety. If there's a problem, for example, we are using the Qatar variety, but is not recommended by the Department of Agriculture because it is susceptible to one disease, blast. When the blast comes, totally they will destroy. So we are now, as a plant breeder, we are trying to develop the disease resistant, blast resistant attacker. So if there is any problem, we will develop a new variety to solve that problem. If there is any problem even that Popeye that side, that's a soil becomes saline, then we will produce saline tolerant crops. So what is our ultimate aim of this course? My courses, crop improvement through biotechnology. Improvement of the crop genetic material. We will try to produce new variety, which is better than the existing variety. Can you understand the English? It's okay, it doesn't matter. We are going to develop a new variety, which is better than the existing parents. If you develop the new Virat Kolumban, that should be better than the Virat and better than the Karta Kolumban and better than the others. So we will develop. So we will do that new variety through two different ways. One is conventional plant breeding, another one is biotechnology, genetic engineering. Okay, that's what we are studying the property. But cell biology, what we are studying, so basic unit at the meantime. Basic unit at the meantime, you can write something here. You have to practice. So what is ribosome? Already you have studied. Within a plant cell, where the proteins are made. Ribosome gives the cytoplasm of bacteria granular appearance in electron microscope. So we will study each and everything slowly. Don't worry. If you want to go for the next lectures, this study you can get it. please remember, I think most of you have studied your A level, you have choose the biology to go for the medical faculty. Most of them, I think more than 98%, maybe one or two, you may get the agriculture. Others, you have tried to be a doctor, MBBS. But there's a problem with finally you got it, the agriculture. Don't worry. But so one thing, you have missed your stethoscope. Nothing else. Remember, mostly you may have aimed to be a MBBS doctor, but now you got it agriculture. Just you lost your stethoscope, nothing else. Otherwise, you can, after just studying this one, you can be anyone, even any secretary or university lecturer or PhD holder, again, you can doctorate or become professor, dean, vice chancellor, even the lot of bank managers. You can see your seniors, including me as your senior. I am the fourth batch. You are 32nd batch. I am the fourth batch. Same AG index number, and I did my this one. Earlier, I also like to become a doctor. Then, but I got agriculture. But thereafter, I finished my basic degree and went for the Japan by scholarship MPhil and did the PhD and become a Dr. Thirukumaran. And I did my PhD in genetic engineering. Then Dr. Thirukumaran, genetic engineer. So I got a doctor and the engineer both. Earlier, even the A level when you choose, you may have the doubt whether you want to be a mathematics or biology, want to be an engineer or doctor. But I chose biology. I lost my stethoscope, but I got it other things. I never lose other than the stethoscope. So we can learn in the next class also. I think it's already time up. Uh, okay, I will stop here and we will learn. Okay, don't worry, you can be a learn each and everything. You can be a anyway. Don't lose your opportunity. Believe that you can be a. Be sincere and hard worker. Even through the agriculture, you can become a. Even as take me as your senior. Now I can put it. Dr. Thirukumaran, genetic engineer, professor in agriculture biology. Even more and more, we have enough time. I will be in university another 20 years. So it's not my end. It's my start. I will grow further. Take care. Thank you very much for your presence, and we will meet on next Friday. You can use the same link. My ID number 45121212. Thank you very much.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.